So today we're going to do the last genetics unit lecture. It's on sex chromosomes and sex-linked inheritance. But I noticed on the quiz, the online quiz, that there were some problems figuring out the pedigrees. So let's practice. Just shout out. What is this? Dominant. Right. So one of the things you need to do in looking at pedigrees is go with your gut. You look at it, it looks dominant, it's dominant. Go back and check, make sure you're right. But usually the first thing you think is correct. This one is recessive, right. And this one is recessive, right. But when you look at this one, you see a pattern. Everybody who's affected is male, right. So this is the segue in today's lecture on sex-linked inheritance. How can this happen? First, we have to talk about what the sex chromosomes are in humans. You probably know already that they're X and Y. Women have two X's, and men have an X and a Y. These X and Y chromosomes share a small region of homology. This is called the pseudo-autosomal region. And why is this important? Well, during meiosis, the X and Y chromosome have to pair up. Right? It'd be like the mules. If they didn't pair up, then you'd have a block in meiosis 1. All the chromosomes have to be paired up in synapses before you can proceed through meiosis 1. So they have this little fake autosomal region. The sex chromosomes are X and Y. Every other chromosome is called an autosome. So they have a little region of homology between the X and the Y where they can pair up and even cross over. All eggs are going to have an X chromosome because females only have an X chromosome to give, while sperm can have an X chromosome or a Y chromosome. The Y chromosome carries mostly genes involved in male sex determination. Half of us in this room are just fine with no Y chromosome. So there's nothing really that essential on the Y chromosome. But if you're going to be male, that Y chromosome is essential. It is what makes a male male. The female sex is the default one. The X chromosome carries genes with essential functions. So you have to have an X chromosome. Everybody in this room has at least one X chromosome. It is essential for life. So the X chromosome carries very important genes and has functions that are unrelated to sex determination. Now, if you're going to think about this in Punnett square terms, you have females that have two X chromosomes. They can only make gametes that have X chromosomes. Males, on the other hand, have two different chromosomes, so they can make gametes that get the X and make gametes that get the Y. So this means that in humans, males determine the sex of the offspring. So poor Anne Boleyn got her head chopped off by Henry VIII because he didn't give her a boy. It was not her fault. So males determine the sex of the progeny because they have two different chromosomes to offer. Now this is just kind of a trivia note, but also it might help you remember some facts about the X and Y chromosome. Sometimes people want to select the sex of their children. Maybe they already have 3 girls and they want a boy, or maybe it's the other way around. One way to do this is you can sort the male sperm to get only Y sperm or only X sperm. Now, how would this possibly work? Well, the Y chromosome is much smaller than the X chromosome. So a gamete, a sperm that has this big old X chromosome, has almost 3% more DNA in it than a sperm that has the Y chromosome. So you can use this fancy machine called a flow cytometer to sort the sperm that have more DNA into a separate pool, because you can use dyes that fluoresce when bound to DNA. The more fluorescent sperm are likely to have an X. The slightly less fluorescent sperm are going to have the Y. So after sorting, they've used a different technique that shows the Y carrying sperm in green. You can see they're pretty pure. So based on DNA content alone, you can get a pretty pure population of Y-bearing or X-bearing sperm. And if you use this approach for just $5,000, you can end up with a girl about 90% of the time and a boy about 75% of the time. So it tips the odds. But anyway, this happens because, you can do this because the Y chromosome is smaller. These are white and red-eyed flies. This is the only picture of Drosophila that I've ever seen that is actually beautiful. I think these flies are beautiful. So I wanted to make sure I showed it in class because in general I find Drosophila very annoying in my kitchen and in my lab because they're all over the place. With X-linked recessive disorders, males are preferentially affected. And this red and white-eyed fly experiment that was done by Thomas and Morgan is the classic example that's used. It was how this X-linkage was discovered. So we're going to go through it here. This is the figure from your book. We start out with a parental generation of true breeding red-eyed flies and true breeding white-eyed males. They're crossed to get the F1. And because the dominant phenotype is red, all of the F1 flies have red eyes. When you produce the F2 generation by crossing the F1s, you see what you kind of expected for a recessive disease. A quarter of the offspring have white eyes. But what's a little bit different is all the white-eyed flies happen to be male. So how did that happen? Looking at it in Punnett square terms can help you figure it out. So in that P generation, parental generation, we had a normal female. It was true breeding red eye. All it had was normal X chromosomes. And an affected male. That was a true breeding white-eyed male. So all it had was a mutant X chromosome to give. So the normal female can produce gametes with normal X. The mutant male can produce gametes with a mutant X or a normal Y. And when you sum those up in a Punnett square, what you get out is all carrier females and all completely normal males. None of these flies are affected. So make sure that you don't get confused. Carrier status and affected are different. So the F1 generation all have red eyes. All the females are carriers. All the males have a normal X chromosome. So now we're going to cross these F1s with each other. And we just said all the females in the F1 are carriers, and all the males had a normal X chromosome. So when we do a Punnett square with these guys, we get our normal 1/4 affected. And those are the ones that got the mutant X chromosome and a Y. So a quarter of the offspring and half of the males are affected with the disease. None of the females have the disease. There's one that has 2 normal X's and one that's a carrier, but none of the females are affected. So in F2, all of the females have red eyes, and half of the males will have red eyes. But don't forget that half of these females are actually carriers and have one of the mutant X alleles. So to see if you understand this, I have a quicker question for you. Adrenoleukodystrophy is caused by mutations in ABCD1, which is a peroxisomal membrane transporter protein. This gene is found on the X chromosome. This is the disease that the kid in Lorenzo's Oil had, if you've seen that movie. It is a disease characterized by normal development in early childhood, but then rapid degeneration to a vegetative state. Children with ALD most likely inherited the disease through their mother or their father. Okay, go ahead and click in. So that's good. Most of you guys get this. The 30% that said dad. If dad carried a mutant X chromosome, he would be affected. So he probably wouldn't be able to be a dad because he would have developed the disease and entered the vegetative state as a child. So the reason why these X-linked recessive diseases tend to be passed through females is because the females don't even know they're carriers. If the male had the mutant X chromosome, they would know it because they would be sick. They would be affected. So the answer is mother. Now, a little interesting side note here. It turns out that the female carriers that are heterozygous sometimes show symptoms later in life. So this would be an example of what kind of inheritance pattern. So the heterozygote is having a phenotype It's sort of an intermediate phenotype. They're not sick and entering the vegetative state of the child, but they're not normal either. So what does that sound like? Incomplete dominance. Right. Yes. So the heterozygote has a phenotype that's intermediate between the normal person and the one that's affected with the disease. Any questions about this? Good. Okay, so hemophilia is a classic X-linked recessive disease that you might have heard about. Queen Victoria was a carrier for hemophilia, and she passed it on to a lot of the allele, onto a lot of her daughters were carriers. Some of these went on to have children with other European nobles, Russian nobles, and all of the males in this pedigree are the ones who are affected. The females are carriers, they don't have hemophilia, which is a disease where your blood does not clot properly. So females carry it, males tend to be affected. Now this is what we call a reciprocal cross, to see if you get it. Now we're going to take an affected female that has an X-linked recessive disease. It's going to produce offspring with a normal male. So instead of the male being white-eyed, now it's going to be the female. So what are the percentage of children affected from that kind of cross? I encourage you to draw a Punnett square. Section A had more trouble with this than I thought they would. Okay, go ahead and pick one if you haven't yet. So we have some problems here. So let's go ahead and draw this one out. So in this case, we have, sorry, I don't have a fat one, so I have the ballpoint. We have a female who's affected. So what's her genotype? XM, XN. It's a recessive disease. So for her to manifest a phenotype, for her to be affected, she has to have 2 abnormal X chromosomes. And then the male was normal, right? So he's XY. Now I said, well, why don't you guys go ahead and draw a Punnett square. So I'll draw one here. This female can produce only XM gametes. You could have made it just half as big if you wanted to. This male can produce X or Y gametes. So when you fill these out, what do you get? Oh. What are these females? Carriers, not affected. What about the males? They're all affected. Okay, so let's go back and look at the question again. So the right answer is A, right? All the boys are affected. None of the girls are affected. They're carriers. So one of the points of this question was to reiterate that carriers are not affected with the disease. You don't see anything until they go on and pass the allele to their kids. Any questions about this? That explanation help? We're good? Okay. So this is not a clicker question. This is a question for thought. Rett syndrome is a developmental disease that causes profound mental retardation, reduced muscle tone, seizures, and autistic behavior in girls beginning at 6 to 18 months. It's caused by a mutation in MECP2, and this gene is on the X chromosome. So Rett disease is only seen in girls. Why is it not seen in boys, as is true for most X-linked diseases? And affected females have one normal copy of the gene. Why do they get the disease? So I want you to think about this, talk to your neighbor, and see if you guys agree on what the answer is, and we'll go over it. see. Does anybody have a good idea why we don't see this disease in boys? Yes, sir. Okay, so suggestion was this is an X-linked dominant disease. That sounds pretty reasonable to me. It's a good answer. But why don't we see it in males? Males have an X. Why is this disease not seen only in boys? Okay, very good. You got me. That's exactly what I said. It's not seen only in boys because it's dominant, so you can see it in female. Why are there no boys? It's not exactly what I wrote. Notice that. Yes?
Does the Y chromosome affect the expression of the X chromosome?
That's an excellent hypothesis. So he said, could the Y chromosome affect the expression of the gene? It turns out to not be the right answer, but could be. Anybody else in the back? So you're saying it's, well, okay. So if you're arguing for haploinsufficiency, right, that you have to have two alleles, it doesn't really work, right, because the guys have one bad one, so they should be sick, right? It's not a bad approach, but I think maybe it will work. Yes?
Could it be because since they're mutated on the X chromosome, what they have, they die?
Yes. Yes. Because they only have one X chromosome and it's busted, they die. The males are not seen because they fail to develop. Embryonic lethal. So those tricky geneticists are going to be doing this to you later in Bio 97. So when you see a ratio you don't expect, one thing to think about is maybe one of the genotypes is just never born. So it's not that it didn't happen, it's just not there to count. Okay? So that's why you don't see this disease in boys, because the boys don't exist. They died in utero. Affected females have one normal copy of the disease. Why do they get sick? It's dominant, that's right. And the reason why they can get by at all is because of X, probably partially because of X inactivation, which is something we're going to talk about in just a minute. So half of their bad X, half the time their bad X will be turned off. And so they can get by with having one normal copy. And here we are, talking about X inactivation. So half of us in this room are in this situation. We have two copies of this essential chromosome. Messing up gene dosage, how many copies of a gene you have, generally causes big problems. Half of us have two of this very essential chromosome, and half of us only have one. So how can that be? How do we fix that problem? Well, the answer is that in females, one of the X chromosomes gets inactivated. This happens early in embryogenesis. I've looked for like 7 years to find exactly what stage of embryogenesis it is that this happens in. I don't know exactly. Early. Something like, 32, 64 stage embryo. Early. And every cell that descends from that cell will have the same X chromosome inactivated. So females are actually sort of a patchy distribution of which X chromosome is active. Which X chromosome gets inactivated is random in most animals. So if you have a bad X and a good X, you can't choose to inactivate the bad one. It's just random which X gets turned off. Whichever X chromosome is inactive stays inactive. The inactive X chromosome is sometimes called a bar body, but strangely enough, it's not completely silent. The figure I found in the literature is about 15% of the genes on the inactive X are still expressed. So it's not totally silent, it's not off, it's just quieted down. There's still some gene transcription. So this cat Ginger here is wandering around outside, and I tell you that orange and black are separate alleles at the coat color locus of the X chromosome in cats. So you see Ginger, and you immediately say to yourself, Ginger is male or female? Okay, click in. 2 easy choices. Good or you don't. All right, let's see what you think. Yay, good job. Okay. Yay. So whenever you see a calico cat, you can impress your friends by saying, that's a female cat. Because all calico cats are female. Because if black and orange are both on the X chromosome, you have to have two X chromosomes to get two different alleles. So tortoiseshell cats, calico cats are always female. If it does happen to be male, then that cat has a chromosomal anomaly and will be sterile, which we're about to talk about in a second. So I said that gene dosage is really important, but you can actually tolerate changes in the amount of sex chromosome that you have reasonably well. And the reason why is because the X chromosomes can be inactivated as bar bodies if you have an extra. And the Y chromosome is kind of small and doesn't have much important on it anyway, so having an extra one of those is not a huge deal. So these are some syndromes that occur in people. You can have 47 chromosomes because 3 of them are X's. These people will be female because they don't have a Y chromosome to make them male. And how many bar bodies will they have? 2, right? 2 in each cell, because they do their dosage compensation by inactivating 2 X chromosomes. Person who's 47 XYY, this person is going to be male because they have at least 1 Y chromosome. These people are relatively normal, but can be a little bit mental disabled, they're tall. Both of these people are going to tend to be sterile because they have the same problem that the mules had. They have an odd number of chromosomes, and when meiosis comes along and they need to partner up with a buddy to proceed through meiosis 1, it won't happen. So most of these people are sterile. Somebody who's 47XXY has Klinefelter syndrome. They also have mental impairment. Somebody who's 45X has Turner syndrome. They tend to be short and phenotypically female. This person will have how many bar bodies? 0, because they only have 1 X and they need to keep it on. You cannot get by without an X chromosome. The X chromosome is essential. You can do without a Y, but not an X. So this also illustrates that inactivating an X chromosome doesn't totally make it silent. Because if you could just inactivate that extra X, this person up here should be totally normal. If that inactive X wasn't doing anything, somebody with Turner syndrome should be totally normal. But those 15% of genes that are still expressed off the inactive X do have some important functions. So how do these strange sex chromosome imbalances occur? Through a process called non-disjunction. I'm going to go through a couple different ways that could happen. So this is a male gamete-forming cell going through meiosis. We're in meiosis 1. The sex chromosomes are the orange and red chromosomes, X and Y. And what happened here is one of the spindle microtubules that was attached to the kinetophore lost its connection. And so when anaphase happens, the homologous chromosomes get pulled to the same pole of the spindle. Because those homologous chromosomes are attached by proteins, cohesive proteins that are important for synapsis. So if the spindle detaches, it'll just go with its homolog to the opposite pole. So at the end of meiosis 1, you'll end up with a gamete here that got both halfway through meiosis gamete that has 2 sex chromosomes. It got both the X and the Y, and another one that didn't get any sex chromosome. After meiosis 2, the sister chromatids will separate. This one will end up making XY gametes. And when they fuse with an egg, which of course will have an X, they'll make XXY people. Down here, this one has no sex chromosome, so when it fuses with an egg, it would make somebody with Turner syndrome. Now you can also have non-disjunction, failure to separate during meiosis 2. So here, I'm showing it for the Y chromosome. The Y chromosome lost its attachment to this side of the spindle, so both sister chromatids went to the same pole. That would produce a gamete that has 2 Y chromosomes and a gamete that has no sex chromosome at all. You can also have non-disjunction at the X in male meiosis 2. That would be possible. In female meiosis, you only have an X chromosome, but you can still have non-disjunction. So here, if you have non-disjunction in meiosis 1, both X's will go to the same pole, and you'll end up making gametes that have 2 copies of the X. If they fuse with a X-bearing sperm, you'll get the triple X. If they fuse with a Y-bearing sperm, you'll get XXY embryos in people. Down here, this one has no sex chromosome. It will make 45X embryos, people with Turners. If this fused with a Y sperm from dad, you have nothing. Because somebody who's Y only would fail to develop. The X chromosome is essential. Now, we said that these sex chromosome non-disjunctions are reasonably well tolerated because the sex chromosomes are special. The X can be inactivated, the Y doesn't have much information. But, I think I went backwards. With the autosomes, this can be a much bigger problem. It can happen with autosomes, and I bet most of you know somebody with Down syndrome because it's not that uncommon. Down syndrome is caused by, among other things, chromosome non-disjunction during meiosis. Down syndrome is trisomy 21. That means you have 3 copies of chromosome 21, often due to non-disjunction. Now, it's not that chromosome 21 is particularly prone to non-disjunction. It's just that people that have non-disjunction of other autosomes tend to fail to develop. It's bad news. So chromosome 21, 3 copies are tolerated reasonably well. Some people are also born that have 3 copies of chromosome 13 and chromosome 18. But pretty much any other autosome, the embryo will fail to develop. You can't have 3 copies of autosomes. It's very bad, except for these 3 exceptions. Monosomy is not tolerated in any autosome. You have to have 2 copies of every autosome. So the sex chromosomes are a little bit special in being able to see these effects of non-disjunction. Does any of you have any questions about non-disjunction? Good. Okay, so we're going to finish up with some independent assortment stuff here. The principle of independent assortment applies equally to all genes. Do you guys think that that's true or false? Okay, look in if you have not. So we're kind of split on this issue. Probably the people who read the book or looked ahead in the lecture chose B, false. Okay? So when does this principle of independent assortment not apply? When the 2 different genetic loci are located next to each other on the same chromosome. So this is a new version of that independent assortment figure. Before, we had Y and R on 2 different chromosomes. And then the dominant R, the dominant Y could be on the same side with the dominant R, or it could be on the same side with the recessive R. But now they're on the same chromosome, so they have to be on the same side of the metaphase plate without crossing over. So when 2 genes are on the same chromosome, they'll tend to go together. It's sort of like they're holding hands. They're stuck together or linked. It's hard to get them apart because they're on the same chromosome. When they're on 2 different chromosomes, it's going to be random which side of the metaphase plate they end up on relative to each other. So we're going to go through an example in your book that shows genetic linkage. Here you have an F1 dihybrid female. So she got this red chromosome from one of her parents. And that has 2 wild-type alleles of the black and vestigial genes. And she got this pinkish one from her dad. And that has a mutant copy of black and a mutant copy of vestigials. And this F1 dihybrid is being crossed, it's test crossed with a doubly mutant male. So a double recessive homozygous mutant there. And what comes out is mostly offspring. They get one of these 2 parental chromosomes from the mother. So most of the offspring are going to get both the wild type black and vestigial genes or both the mutant black and vestigial genes. If they were sorting independently, these would get mixed up. So let's go through the next couple of slides to see why we don't get these guys sorting independently. If you just walked into this problem and didn't know they were on the same chromosome, you'd look at this dihybrid phenotype and you would think that each of these gametes is going to be formed with equal frequency, right? It's just as likely that this wild type B will go with the wild type vestigial as the wild type B with the mutant vestigial. If that were so, this test cross individual can only make homozygous, or can only make the recessive gametes with the mutant black and the mutant vestigial. So you could make a Punnett rectangle like this, and you would expect all 4 of these phenotypes at equal frequency. If the alleles were assorting independently, they were on separate chromosomes, sorting independently, all 4 kinds of flags would be seen with equal probability. But what actually happens is that the parental type offspring, the ones that got the parental chromosome, are much more likely than the recombinant offspring. Why is that? Well, the parental chromosome can just be inherited as is. The only way you can get the recombinant offspring is if a crossover occurs between the genes B, black, and vestigial. So black and vestigial chromosome is a linear piece of DNA. If black and vestigial are very close to each other, the likelihood that a crossing over event will randomly occur between them is low. But if black and vestigial are very far apart on that linear chromosome, there's a good probability that there'll be a crossover between them and they can exchange between the 2 parental chromatids. So these are recombinant chromosomes because a crossover event occurred between the 2 genes. Well, these are the parental chromosomes. No crossing over occurred between the 2 genes. These parental chromosomes are present at a higher frequency here because the black and vestigial are relatively close together. So recombination only happens, so 17% of the offspring are recombinants. If these genes were right next to each other, you might get 1% recombinance. If the genes were very far apart, you might get 50% recombinance. So that would be just like they were sorting independently. The farther apart these genes are on the chromosome, the more likely a crossover will occur. Okay, so let's see if that makes sense. Because the traits Mendel studied assorted independently, the loci are on different chromosomes. Is that true or false? Okay, so then if you have not. All right, well, the answer is false. Okay, so 2 of the traits that Mendel studied are now known to be on the same chromosome. But they're far enough apart on that chromosome that they appear to assort independently. The recombination frequency is high. Okay? So I finished earlier in section 8. So if you want to ask me questions, please do. Otherwise, start your holiday weekend early and enjoy it. See you Monday.